Okay, so why I show you that? Because I didn't learn biology like that. I learned that this is a diagram and nobody thought that that moved. And she has an entire lecture about all the functions, new functions that it can have because they move. And without live living imaging living samples, we didn't know that. Yeah, so there's a lot of things that we don't know about the cells until the GFP, all these fluorescent proteins appear. So the biology maybe that you learn now will be very different to the one that your kids will learn because this is changing very fast. Okay, so the key idea that I wanted to show you is that similar to our cities, yeah, although uh, in the picture things are not moving, the endomembrane system is moving a lot and it's very dynamic. And these intercellular membranes that have the endomembrane system, they need to be constantly renewed, and the ER is responsible of renewing both the proteins and the lipids. Okay, so I missed this. What is the endomembrane system? A system of interconnected membranes within the eukaryotic cell. You already know that because you have seen it. Exceptions are the mitochondria is not there, the peroxisome is not there, but the Golgi apparatus, the lysosomes, they are, and maybe sometimes in your book they put the nuclear envelope there. Those are examples of interconnected membrane organelles. And there is a lot of traffic, and so I'm going to show you not all of them because it will be super complicated, but I'm just going to show you the smallest one of those ones, okay? That it will be a lysosome. So there is something here that is very important that maybe some of your articles will talk about that is called vesicle trafficking. Yeah, similar to the cities that I showed you before. The cells have a lot of traffic, and traffic of what? Lipids and proteins between organelles. Yeah, and these have to be very regulated. So here they are showing just the traffic of a lysosome. Yeah, and look, there are things that are you have uh, traffic of lipids in red, proteins in pink, and here you have carbohydrates, and they go through endocytosis and form an endosome, and they go to the lysosome. Okay, and they go outside of the lysosome and go outside of the cell through exocytosis. Okay, and if you see the lysosome, you remember that before I just show you a bilayer here, they have also enzymes and channels like the membrane that we generally talk. So they have ion channel transported, they have exported, they have a lot of, we generally just put a circle there. This is organelles are much complex, and they are catalyzing all these things. They also, there is traffic to autophagy. So endocytosis, they also have autophagy here that is breaking the organelles, the cells is eating. And also you can, some of those things from the lysosome can go to the Golgi apparatus. Okay. So again, I just wanted to show you that even the simplest organelle, there is a traffic of a lot of molecules. And if we go through Golgi or the endoplasmatic reticulum, they will be even much more things. Okay. Oh, where we were in Zia, yeah. Okay, could you read this one and help me to answer it? You say B, yeah. So direct, now we know that A will be for the next test. Incorrect. No, definitely mitochondria is not there. Ribosomes, they don't have membrane, so no. Peroxisomes, we already say no, yeah. They come from a different origin, so perfect. So these are the four of those one, and sometimes if you check your textbook, sometimes they can include the nuclear envelope because they are connected. Do you see that I will show you in a second. A picture they zoom in here, and do you see that they are connected here? They have many connections, so this is why sometimes they include it there. Again, as a reminder, peroxisomes are not there. Okay, perfect. Golgi apparatus and lysosomes. Thank you. Noella, could you tell me the function of the endoplasmatic reticulum and the Golgi complex? What they are doing, those two? Mm-hmm. Mm-hmm. Perfect. So both, like all of these organelles, are producing or lipids or proteins are, and they are packing it and storing it. Perfect. The Maddie, I don't know if you know this one. I didn't talk a lot about this one. Do you know what is the so Noela, sorry, tell us about this part. Now we're talking about the endosomes. Do you know what is the function of the endosomes? Do you remember? Yeah, they transport the molecules during the cell, and some of them they can be modified in different sections, but here they do too. And Jenna, the lysosomes here in yellow, do you remember what they are doing? Perfect. Yeah, they are digestion. Oops. So the answer, yeah, it's okay. There was a missing in one of the slides here. So perfect. The Golgi apparatus is in the synthesis of proteins and lipids, and the sorting of those one. The endosomes carry the materials, and the lysosome is digesting all those things. Uh, Sorry. And now, okay, I already showed the answer. It's okay. How does the endoplasmic reticulum contribute to cellular transport? By synthesizing and transporting vesicles to carry it to other parts of the cell. So you already knew that it was for the test. Okay, so endosomes. In the previous class, it was just the introduction, so I didn't talk a lot about the endosomes. Here again, I'm going to just show you that the endosome, here you have the plasma membrane. This is a representation of a cell. Here you have the Golgi apparatus, the Golgi complex. And what is happening is that you have these vesicles, yeah, that form as a result of the endocytosis. So here you can see that they come from the endocytosis. They can also, they have some molecules that come from endocytosis, or ones that they will go back to recycling. That is very important. Again, as I will show you in few lectures, one of the problem, like why people, for example, a basketball player, a volleyball player, when you get injured and they have medication for pain, there is a point that the medication don't work anymore. Yeah, why it doesn't work? Like so we have soccer players. One of my favorite soccer players from Colombia, got injured so many times that no matter how much medication they put it, he doesn't he still feel pain. Why? Because there is a receptor here for pain. You know, and those receptors they need to take time to recycle. Yeah, so there's a lot of cell biology related to the pain. If you don't have those receptors, or you cannot block those receptors, there will be problems. And this is also related to opioids addiction, yeah, because this is, you are controlling also pain. And I know that I didn't say completely the story, because that will be a completely story that I will tell you in a few weeks. Okay, but important endosomes, very important molecules that they can get some nutrients or some molecules from the endocytosis, and they need to recycle them. They can also mm, I'm sorry, Eric, you don't know what is this word. What do you think is transcytosis? Another weird thing. Yes, that is another word that it referred to endocytosis, all this molecule movement. But here in this case, you see that there are some that are go here, and here this one is moving to another cell. So transitosis, in this case, there is also molecule movements not only with the lumen, yeah, outside of the cell, but within the other cells that they are supposed to have proteins junctions that don't allow that, or they can go to the blood cells. This transport is different, so the endosomes can also go through that. Okay, so here I just wanted to show you the story. What is an endosome? And here I wanted to compare with a lysosome. And remember, the endosome is a vesicle that formed from the invagination, from a pinching of a cell of a membrane, and that they while the lysosomes are an organelle that have degrade enzymes that degrade organelles, and the endosomes mainly form during the endocytosis. Okay, so in case that you wanted to study, I know this is a lot of terminology, but this is something that I didn't talk last time. Quickly, another video. Okay. Okay, so here, in case that you have to study, I put all the material in case I know that some of you study from the slides. If you want to have for the next test a higher grade, you can also read something from the textbook. I try to put a lot of questions from the slides, but some of them also come from the textbook. Okay, so this section in this 20 minutes, I told you the function of the year, that is the flow of materials that you already know. And all of you told us that I think all of you know that there is a transport of vesicles, that they have proteins and lipids, that they go to all these different organelles. And I wanted to emphasize that they were very dynamic. So I show you many movies, okay? That is not like this, that is very dynamic. Okay, now I'm going to talk about the rough ER. And again, I'm going to talk about these different topics in case that you want to study later for the test. So key thing that I'm going to tell you in the beginning of this, I'm going to tell that you're going to identify the different components. Some of them will be very easy. Some of them will be a little bit more complicated. So you will have to identify them here. Then I'm going to tell you some of the functions that Noella already told us some of those. I'm going to show you some of this. For the test, Rico, do you see this picture here? This is the process of translocation. Ribosome, do you see the ribosome here? And look, it produces the polypeptide, and the polypeptide go inside of the ER. Okay, so you will have to identify the 4 steps. This is what Noella told us, the biosynthesis of lipids. And I don't know if you have heard about chaperone proteins. If somebody knows what is a chaperone protein. They're very famous, very important for curing cancer. So we will talk about those one. Chaperone proteins that are very important. Here I already say folding of proteins. This is helping the proteins to get folded, and the ER is the rough has many of those ones. Okay, so we were Duncan. Pretty easy. What is number one? Nuclear power, and this is why I give you slides in case that so you are paying attention and you can, and if you remember, hello, welcome. Uh, Asher, number two, what is that thing? Duncan, you're a best friend ever. Thank you. Try to number three, tilt more the computer. Oh, he also good smoothie air, and because there is no ribosomes. Um, Number four, oh, same question. Oh yes, that is not the rough ER. This is the external space, and you're checking the slides. Uh, Number five, uh, Adriana, do you see a computer of Derek? Number five, what do you think is there? The one that's with T. I'm just oh, oh, I'm also dyslexic. Okay, so I understand. Number five, Felicia, help us. Yeah, so inside the direct tolerance, we have the lumen or the internal space, and here in number five we have the external, so we have the cisternae. Pretty good, yeah. Zia, help us with numbers. Okay, there is no number six. I'm sorry, there is no line, or I delete the line. So we are going to skip number six and go to number seven, the ribosomes. Noella, no. Okay, so pretty easy. All of you know there might be a question like this in the test. Okay, and it will be pretty easy. Okay. Okay, why we call it endoplasmic reticulum? So endo, inside, yeah. So inside is it's inside of the cytoplasm, and reticulum mean networks, and they are huge, yeah. They cover more than 50 or from 50 to 90% uh, of the space of mammal cells, yeah. And I will show you that in a second. And here again, these are the two things that you need to remember. The cisterna, that is the thing outside, and the cisternal space, or the lumen, the ER lumen, okay? So when we see these pictures, we see that there is a network, yeah, and I will show you more pictures of that, of sacs and tubules that are associated with vesicles. Here you saw the nuclear parts, and you see that they are composed of many proteins. So if you choose an article that talk about that, I'm just going to show you a schematic that is like a tube. You have to imagine that is much more complex like that than that. But for this class, we are just going to call it nuclear pore. I already told you about the name. And remember, I just wanted that you remember that there are two key components. That is the internal, yeah, and the external component, okay? So you can, in the next slides, for example, to explain you what is the translocation, you need to know what is the ER lumen or the cisternal space. That is the inside of the ER. Okay, so this is a cell that here you are highlighting all the ER. So this is why 50 to 90% of the membrane in mammalian cells is surrounds the ER lumen, because it's just huge. Yeah, the ER is inside, and it has and there is a lot of ER. So For that reason, if you watch all the video lecture of the person that I show you, it will show you has so many more functions than we expected. Okay, I think I am confused about where we're. Oh, I think we were here. Noella. So I wanted to show. Did I say Noella? Yes. Good. Getting closer. Noella. So here you can see the nucleus, and here are the chromosomes. And here, how many membranes do you see here? 2 membranes. Derek already told you which organelle has 2 membranes. Which other organelle had 2 membranes besides those one? That is in plants. That is chloroplast. So there is a lot of hypotheses about that the nucleus could be another example of endosymbiosis. However, we cannot test it very well. So right now we are completely sure that the mitochondria and the chloroplasts are examples of endosymbiosis. We just don't know why the nucleus also have not only one membrane, but it has 2 membranes. We just know that to have 2 membranes, it allows them to traffic very well the enzymes. So the enzymes, they are not going to go inside. It also it allows them to protect the genetic material. So we don't know why this organelle evolved 2 membranes, the nucleus. And you will see a lot of hypotheses in the future. But it's just fascinating thinking that maybe even the nucleus could have gone through endosymbiosis, but we don't have enough evidence to talk about to be sure about that. Okay, so here is the nuclear pore. This is a complex of proteins, so the RNA is going to go here, and here I wanted to show you this is the ER cisternae that Felicia told us here, and here we have the ribosomes, yeah, and if you see all these green things are connecting the ER through to the nucleus, yeah. So instead of being 2 independent organelles, they are connected. There are 3 components that I want that you remember: the nuclear envelope that is in blue, the ER tubules that the lady was showing you that she was filming, yeah, and here the ER cisternae. Okay, so the inside of this. So again, rather than 2 disconnected organelles, they have like highways, so the messenger RNA go there, and here the ribosomes are ready to do the translocation. So remember, the nuclear envelope is the most or visually domain from the ER, yeah, and they are great. This double membrane is great to selectively control the transport of molecules. So I told you they are nuclear ports, and I'm not going to ask you in the test all these additional proteins. Yeah, but there are many proteins that are involved in the traffic of the nucleus. Okay, so nuclear envelope, very important. Matt is super easy question. I don't know. Could you help me with this question? Read it. Lipid synthesis and detoxification. Okay, so we will talk about that in a second. I will show you how this happened here. So I already told you all the components of the ER, and you were able to identify all of them. I show you that I zoom in the nuclear envelope, and I show you there are many tubules. I show you that the nuclear envelope have 2 membranes. Now I'm going to show you some of the functions of the ER. Maddie already told us 2 functions of the smooth ER. Now we're going to talk about general functions of the 2 ERs. So we have one function related to proteins. That is, the proteins are formed, unfolded in the ER, and they can be sent to the plasma membrane or to different organelles like the mitochondria. In addition, some of those proteins can be exported from the cell. So if you took anatomy and physiology, there will be a lot of proteins that they can go outside of the cell. And later we will talk about some examples of some hormones that they are sent there. And some of those hormones could be lipids, and some of them can be proteins. Okay, so two things that are the biosynthesis of proteins, and they will be put it in the membranes, plasma membrane, and in the organelles, and some of them they will be exported outside of the cells, like what happened with hormones. The second thing that could ER do, yeah, and the enzymes of the ER is related to replenish all the lipids of the membranes in all the lipids in the different membranes, yeah, both lysosomes, plasma membrane, all these different organelles, and also to produce different lipids for hormones, for other needs, to store energy, other different sources of lipids. So two things: biosynthesis of lipids, and these are some examples. And another one is the source of lipid to assemble to form intracellular and plasma membranes. Okay, so four functions. And later I will show you about this lipid synthesis. I just show you the lipid synthesis, and then I will show you the detoxification. Jenna, where do you think that detoxification occur in the organelle, in the body? Sorry. Yes. So we'll talk about that when people. So, for example, if we have 10, I don't know, 5 years every day, we will have a lot of ER in our liver. Yeah. But this could have some side effects too. Okay, so do you know this? There are two type of ER, the rough ER and the smooth ER, and you know this. These are all the functions of the ER, yeah? And in the following sections, I'm going to talk about some of those ones. I'm not going to talk about, like there are just so many, and we will, in the future, there could be even more. So, for example, the smooth ER has the lipid biosynthesis. I already told you that they export unfolded proteins, and we will get talking about some of those ones. I'm going to talk about the protein synthesis, and you know all this information. I just put it there in case that you want to study. But now, Eric, sorry, unlucky. This is the process of translocation. Yeah, I'm sorry, it's not. I don't. I think there is a little bit there about that, but I add these slides. Based on this figure, if I put this in a test, what do you think that is translocation? If you have look at this. Okay, so there is a ribosome
The
there.
movement of some receptor
Okay,
or signal particle.
okay, so here you have the ribosome, and what is happening in the ribosome? Translation. Yeah.
Okay.
So you see that the messenger RNA is leaving, and now you have translation, and instead of going outside of the cell,
Oh, it's putting it in the cell.
yeah, that is the process of translocation. And as Eric told us, there are different molecules involved into that. Yeah, that they work like as Legos, that you attach one to the other one and allow them to attach to the ribosome properly. Okay, so just for the test, remember, translocation refers to the process by which newly synthesized proteins are transported across or inserted into the membrane of the ER. So some of them here, they were inside of the ER. Some of them can be even in the membrane, depending on they are soluble or not. Duncan, what is the first step based on this figure? What do you think is step number one? What is the first thing that happened? Sorry, I don't have that in the slide. Next time I will add it. It's only the picture. What do you think from there? What do you see that is happening in the step number 1? There is green thing.
The green thing's like signal.
So it's a protein that is going to attach to the signal sequence. That signal sequence is the first part of the translation, yeah, and it's going to attach there. Very important. So first thing, what happened here is the signal sequence recognition that they attach they're going to the ER, so they're going to attach to the N terminus, and this is an important, this SRP, the green thing that you see here, is very important because it leaded to go to the ER. And again, second thing that it will happen is that the green thing, the SRP, is going to dock here in the ER membrane. So this SER is very important because it's going to attach all the ribosome here, and here you are going to have this protein that is called translocon, that is just a channel that it will allow the the protein to go inside of the cell. Okay, so here you find the translocation through the translocon. Yeah, that is a protein that conduct to the ER. And later we will talk about this sec. And here is all the information. And finally, the last step is this one, when this orange protein, that is the peptidase, remember all the enzymes have this ending, so it's going to cut these initials, the sequence initiation, and now the ribosome can leave and the protein is inside of the cell. So what is the translocation is the process to put the newly synthesized protein inside of the ER. How? There are 4 steps. There are different proteins involved that allow to guide the ribosome to the ER, to attach it, and to that the protein go inside of the ER and is properly folded. Okay, so I will add these slides later if you remind me, and you can study this too. This is also in the textbook in case that you want to read it. Okay, so I told you that the ER has multiple functions. One of them was a new topic. Now you know replication, transcription, translation. The next step is translocation. Proteins go inside of the ER. Other functions, I here other functions here is if you remember here, there is a peptide chain. And last time I told you there is here, do you see a sugar? And here is joined by the end link. Yeah, so this is very important because it can lead the protein to the specific steps, and this process is called glycosylation. So this is starting the ER and continuing the Golgi apparatus. Another process that is happening here. Do you see this protein that is here that have this that is going to fold and is going to form these disulfide bridges? So those happen in the ER. Okay, so those are more functions that are in the ER. And finally, very important for cancer, why sometimes people can get cancer, so many reasons. One of them is that the proteins, they don't have even mutate, the DNA doesn't have even mutations, or they could have mutations that affect the way that the proteins fold. And if the proteins don't fold properly, they or don't work or work too much, and they can cause excessive proliferation or lack of cell death. So for that reason, the body developed these proteins that are in charge of help to properly fold. So here, this is a diagram that shows a polypeptide, and of course, this is just a diagram to make it easier. The the chaperone is not going to look like this green uh, green container, but the idea is that it goes inside and it helps it to correctly fold it. Okay, so chaperone, it prevents misfolding. It also can, here, this is a diagram that shows that if there is an incorrect misfolding, this type of chaperone called pad 10, it can replace the wrong place where it was misfolded and put it in the right way. So again, it's repairing the proteins to fold properly. Here is actually a diagram of a chaperone protein. This is why I showed the first one that it was very schematic. Here you have the ribosome, the translocation. Here is the protein called translocon, and here you have the polypeptide, and here you have the sugar that we talked in the previous slide that it add in that area. So here we have this protein called calnexin that is involved in helping to attach to the sugar and allow the protein to fold it properly. Okay, there are hundreds of chaperone proteins, and this is just an example. Okay, so I told you about the recognizing and removing misfolds, assembling multimeric proteins, and there is more information that you can find there in your textbook. Here, this is one of the famous one. Okay, so we already talked about the endomembrane system that I'm sure that you already all know. I told you about some of the different components of the ER, and I told you the different functions of the ER. Yeah, there are many more. I just covered some of those. Here is the topics of the smooth ER. I don't think that I will cover all of them, so you can give it a look in case that I don't cover them, all of them. But I'm going to tell you that the smooth ER is different because it doesn't have ribosomes. This is a picture of the smooth ER, and is located especially in the liver and in glands like the testes and the ovary because they produce hormones. And we will talk about some of these functions. So we have talked about few of them in the previous section of this of the rough ER that are in green, and in blue you see some of the function of the smooth ER. So here you can see a picture of the smooth ER. And the smooth ER is located in all the cells, but especially in the liver. Why? Because they need to be there is where there is a lot of detoxification, and also there is where you have the storage of some of the carbohydrates and lipids. So it's very important the ER has a function there. And also, I will show you two examples of these two, the testes and the ovary, that have also big ERs. And in particular, the smooth ERs. So all of this, we will cover them later, some of them. Okay, where were we? Okay, Asher, sorry. Do you know what is this? Aldosterone, cortisol, estradiol. Have you any of those things? What are those molecules? Uh, yeah, and they can work. They are involved in the regulation, yeah, of our body. Yeah, they can work also as hormones, yeah. So those one are produced in the testes and in the ovary. So to do that, if you see all of them are they will all of them are lipids, so they are required. They need to have a big ER there. I already told you about the liver. In the liver, So as I said, when somebody drink too much, you know that oh, you can help me with this, Angus. Why do you think that when somebody drinks so much, I told you that they need to detoxify that alcohol and it happened in the liver, but why it destroyed the liver of the person? If you have somebody, if you know an alcoholic, later the liver will be all destroyed. Why do you think that that happened? So it will start developing so much ER that then the enzymes and they will have so many enzymes and produce so many extra products that those one will end up damaging the entire liver. So the ER is very important, but there is a limit. Okay, and I will show you. This is what we call detoxification. So the although it does a wonderful job detoxifying our body, what it does is take something that is very harmful and turn it into less harmful. It doesn't completely prevent the damage, and it also turns something that is harmful, even toxins, into something that it can be eliminated. Yeah. So when it's overloaded, there is a lot of stress. You can have a lot of problems. And again, if you are taking if you took cells and energy, or you are remembering what is a polymer and monomer, so the liver is in charge of breaking all these sugars there, and it has an enzyme to do that. Okay, so I told you about the detoxification and the carbohydrate regulation. Okay, so almost done. In the following lectures, this ER and endomembrane system is going to be heavy classes. I'm going to show you that the smooth ER is very important for the post-transcriptional modifications. So they have the process of glycosylation. So you show the cells the vesicles where to go. If you have the right sugar, you will go to this direction. If you go to the wrong sugar, the vesicles can get lost. Another thing that they do is they produce the disulfide bonds too. Okay, so we have talked about some of these functions. There are more. Now I'm going to finish the class, talk about two more functions. So one of them is this related to the calcium storage. I know that you don't. Do you remember how it's called of course you remember because you have the slides. If you go to the next slide, what is the name of the endoplasmic reticulum of the muscles? The endoplasmic reticulum of the muscle has a specific name that I will tell you in a second, but oh, I already told you about this. Sorry. Sorry, I skipped something. Later, I will talk about the muscles. This is what I told you about the drug detoxification. They become so you have drugs or toxins and they make it less harmful or easier to eliminate. If you look in your textbook, this cytochrome P4050 is the most important protein involved into that. What they are doing is, as I said before, they are modifying these molecules. And in this case, you see that there is an OH. So this is called hydroxylation. So they just modify the molecules to make it or less harmful or easier to eliminate, in this case, to make it more stable. So you have a drug here in red, and you put an OH and do this kind of for enzymatic reactions. I told you that this is called hydroxylation, and this is why they are prominent in the smoothie R. Okay. Already you already know this, that the smoothie AR is involved in the detoxification of drugs and metabolizing of carbohydrates. Oh, sorry. I think my brain already skipped. I was so excited because I thought that we were almost done, but there is a few more things. So I already told you there is the regulation of the concentration of glucose. So I already told you that there is a polymer that turns into a monomer. So we are going to turn these long glycogen molecules into glucose. So we have a specific enzymes to remove this the phosphate group to the glucose, and this happens because there is an enzymes in the smoothie R, and those ones are found in the liver. So it turns the polymer that is the glycogen into the monomer that is the glucose. So this is what happened. Okay, Angus, before I fall asleep, you help me here. What is happening in here? Will you tell me in this diagram what is happening? You have this glycogen granule that have a lot of sugar that are all together, a lot of glucose. So you had a polymer, and you need to break it into a small. So here you have a sugar that has a phosphate, and to remove the glucose, you need to go to the ear, and it remove the phosphate group, and then the glucose can go outside without the phosphate group. Okay. This it could be a question in the not in this test, but in the test, but also this glycogen important for cells and energy. Okay, so we go from a polymer called glycogen to a monomer that is the glucose, and all of this is happening in the ER. Next class, I will tell you very quickly that the calcium storage take place also in the ER. And this guy here, this Moutier, has a sarcoplasmic reticulum because it's a slightly different type of ER. And you can start taking the quiz. But before you start, remember, tomorrow we are not going to be in the lab. We're going to be downstairs. And you can, if you want to leave earlier, read your article and start making a summary, okay? Or the article that you're going to show in the poster, okay? And tomorrow later I will finish this. I think I answered all the questions.